MORFlux is our tool for building and simulating metabolic models entirely in the web browser, allowing predictions about the metabolic flux of an organism to be made and metabolic engineering interventions to be tested in silico. MORFlux uses FBA, or Flux Balance Analysis, a modelling method that relies only upon knowing the suite of genes in an organism and the metabolic reactions that they code for. This set of metabolic reactions are represented as balanced equations and solved mathematically using linear programming with a particular objective being optimised. The objective is usually to maximise the biomass production and hence growth of the simulated organism. Constraints can be applied to each reaction to limit the uptake of a carbon source to the network, for example. MORFlux allows you to upload models in a number of common formats, and there are also pre-loaded models to work with. In this example, we're going to use the IJR904 model of Escherichia coli. An FBA model is composed of three main parts: a list of reactions, a list of genes, and a list of metabolites. And there are separate tabs for each of these in MORFlux. Once a model has been optimised or solved, the predicted fluxes for each reaction appear in the Fluxes tab, and the amount of flux through the objective is also displayed in its own tab. The following example is based on a publication from Professor Sang-Yup Lee's group, which uses a metabolic engineering approach to produce L-valine in E. coli. By selectively knocking out genes to remove competing pathways, they were able to channel flux specifically through the valine biosynthetic pathway to synthesize this commercially valuable amino acid. Starting from the predefined model of E. coli IJR904, we can make the successive changes in our network model as the authors did experimentally. First, to prevent the model from taking up valine or exporting it, we'll set the bounds of the two valine transport reactions to 0. This initial model has a biomass flux of 0.92. Let's call this 100% biomass. We can force the model to produce 10 times more valine by increasing its stoichiometry in the biomass reaction tenfold, from 0.402 to 4.02. This drops the biomass flux to 0.7, that's 76% of the original biomass. Next, we'll simulate the removal of competing pathways for leucine, isoleucine, and pantothenate production by setting the bounds of the highlighted reactions to 0 using the method shown previously. This should channel all of the flux from 2-keto isovalerate through to valine. The biomass flux drops to near 0. To compensate for the loss of leucine, isoleucine, and pantothenate, we'll add transport reactions to allow small amounts to be taken up and used for the cell's biomass. This restores the biomass flux to 0.74, or 80% of the original biomass flux. Our modelling in MORFlux has shown that we can overproduce valine at 10 times the cell's required flux, with only a 20% drop in biomass flux, from 0.92 to 0.74. MORFlux lets you carry out complex metabolic modelling entirely in your web browser and to test metabolic engineering interventions quickly and cheaply. Find out more about MORFlux at morf-db.org.